Hello, this is Sarah LeBrun-Blaschka, Director of Education at Holistic Matters. And today I'm here with Dr. Frank Hu, MD, PhD, who's going to give us an interesting talk on epigenetics benefits of healthy diet, lifestyle, and aging and longevity. Dr. Hu is a chair of the Department of Nutrition and a professor of nutrition and epidemiology at Harvard T.H. Chang School of Public Health. Today, I want to welcome Dr. Frank Hu, and I'm excited to hear your presentation. So Dr. Hu, whenever you're ready, let's get started.
Thank you. It's my pleasure to give a presentation on epigenetic benefits of healthy diet and lifestyle, aging and longevity. This is the outline of my presentation. First, I will briefly describe the epidemiology of human aging, and then I will discuss the role of diet lifestyle factors in age-related diseases and longevity. In terms of epigenetics, I will discuss the effects of diet and lifestyle factors on epigenetic changes, and then I will discuss the associations between epigenetic modifications with age-related diseases and longevity. And then I will discuss novel biomarkers of aging using epigenetic marks. In the past several decades, human populations have experienced rapid economical development, nutrition transition, and substantially improved longevity. However, aging is the inevitable outcome of human life. Aging is related to progressive decline of physical, psychological, and social functions, and significantly increased risk of chronic diseases such as cancer, cardiovascular disease, neurodegenerative diseases, and also increased risk of death. So to slow down or reduce the aging process, our goal is not just to live longer, but also live better. Many factors can influence human aging and longevity, including our genes, diet, lifestyle factors, our environment, and also epigenetics. Epigenetics integrates the influence of both intrinsic and external factors, and to a large degree reflect the interactions between our genes and our environment. There is convincing evidence that a healthy diet and lifestyle can reduce risk of chronic diseases and improve longevity. In the past several decades, we have conducted extensive analysis on several lifestyle factors, including physical activity, not smoking, moderate alcohol consumption, eating a healthy diet based on alternative healthy diet eating index, and maintaining a healthy body weight. In our cohorts, we found that people who adhere to a healthy diet and lifestyle have significantly reduced risk of chronic diseases. In particular, people who adopt a low-risk lifestyle had almost 80% reduced risk of coronary heart disease, 90% reduced risk of type 2 diabetes, 40 to 60% reduced risk of cancer, and 40 to 50% reduced risk of stroke. This data indicate powerful impact of healthy diet and lifestyle on the reduction of the risk of chronic diseases. However, only 3 to 4% of the participants in our cohorts followed a healthy diet and lifestyle. Recently, we estimated life expectancy at age 50 according to the number of low-risk lifestyle factors in the U.S. population. For a woman who didn't have any of those low-risk lifestyle factors, their life expectancy at age 50 was 29 years. However, if a woman who adopted all 5 low-risk lifestyle factors, her life expectancy at age 50 would be 43 years. So the difference between those two groups of women was 14 years. For a man who didn't have any of those low-risk lifestyle factors, the life expectancy at age 50 was about 26 years. However, if he followed all the 5 low-risk lifestyle factors, his life expectancy would be 38 years. So this study indicates that following a healthy diet and lifestyle can substantially improve life expectancy for both men and women. Despite convincing evidence on the benefits of healthy diet and lifestyle factors on longevity, the biological mechanisms underlying the health benefits of a healthy diet and lifestyle are still not completely understood. In recent years, epigenetics has emerged as a potential mediating mechanism underlying the association between diet and lifestyle factors with aging and age-related diseases. In this hypothetical causal diagram, our diet, lifestyle, environment, and our genetic variations can influence epigenetic changes, which in turn influence intermediate phenotypes such as inflammation, and eventually that can influence our aging process and age-related disease risk. So what is epigenetics? Epi is a Greek prefix, means above, over, or on. Epigenetics are modifications on the genes that can lead to changes in gene expressions and functions that do not depend on the DNA sequence. DNA methylation is a common epigenetic mechanism that occurs by the addition of a methyl group to DNA. Epigenetics has several features. First, epigenetics can control our gene expression and functions because certain epigenetic factors can cause some genes to be turned on or off. Also, epigenetics is everywhere because our diet, exercise, sleep, smoking, environmental exposures, and the disease status can induce epigenetic changes at certain genes. Epigenetics makes us unique because the different combinations of genes that are turned on or off by epigenetic factors is what makes each of us unique. Epigenetics is reversible even though we cannot change our DNA sequence. Finally, epigenetics can be passed on to the next generation. So that's why maternal diet can impact epigenetic changes in offspring and also future disease risk of the offspring. 3 general approaches have been used in DNA methylation studies. The first approach is the candidate gene or gene-specific approach. Candidate genes are typically chosen based on the literature or priori knowledge. And the epigenetic changes on those genes can be associated with risk factors or health outcomes. The second approach measures global or average levels of methylation across the genome. And association analysis can be conducted to test whether the methylation levels are associated with health outcomes or disease phenotypes. And the third approach is called epigenome-wide approach or EWAS approach. This is very similar to genome-wide association studies or GWAS. And this approach is an agnostic approach that measures numerous epigenetic marks across the entire human genome. And EWAS is typically using a technology called methylation chip, and the Illumina human methylation 450K chip can measure 450 methylation marks across the human genome. And the newer Illumina methylation EPIC chip can measure more than 800,000 methylation marks across the human genome. There is convincing evidence that diet and lifestyle factors can influence epigenetic changes. Dietary intakes, especially intakes of micro donor nutrients such as folate and B vitamins, can have a direct impact on DNA methylation, gene expressions, and health outcomes. This is a study published in Clinical Epigenetics in 2015. In the figure, it showed changes in DNA methylation levels in the placebo group and B-vitamin supplementation group in the 2-year supplementation trial. Compared with the placebo group, individuals supplemented with folate and B12 for 2 years showed a significant increase of DNA methylation levels at many genes, and some of the genes are involved in B vitamin and homocysteine metabolism, which is relevant for cardiovascular disease. There is also convincing evidence that maternal diet during pregnancy can influence DNA methylation patterns of the offspring at birth. This study showed that maternal folate status in pregnancy was correlated with methylation alterations in newborn cord blood in 2 European cohorts. This study identified 58 significant CPGs, and the biological functions of some of the top differentially methylated genes include folate biology, neurotropic defects, neurological functions, or various aspects of embryonic development. This study provides evidence that maternal folate status may influence the developing epigenome of the offspring and the fetal programming of the child for future disease risk. There is promising evidence that diet may be able to reverse the adverse methylation alterations in the genome. In the crossover randomized trial, exposure to PM2.5 induced methylation changes in genes involved in mitochondrial oxidative energy metabolism. However, supplementation with B vitamins prevented or mitigated these adverse changes. In the dietary prevention trial, adherence to Mediterranean diet enriched with olive oil or nuts was associated with methylation changes in inflammation-related genes in peripheral blood cells. In this study, 8 genes related to inflammation and immune modulation were analyzed for methylation changes. There is strong evidence that cigarette smoking can influence DNA methylation. In the U.S. study of smoking among 600 Chinese participants, we identified more than 300 CPGs that were associated with smoking. Each of the CPGs were associated with expressions of their located genes. The most reproducible locus in the U.S. study was located in the gene called AHRR, and this gene is involved in metabolism of a harmful chemical compound in cigarette smoking, which is polycyclic aromatic hydrocarbon. And this study provides evidence for the epigenetic mechanisms underlying harmful effects of cigarette smoking. There is also evidence that obesity can influence DNA methylation. This is a very large U.S. study published in Nature in 2017. It included more than 10,000 participants and identified more than 180 methylation sites, which were significantly associated with BMI. So one question is whether the methylation is the consequences of adiposity or the methylation causes BMI changes. The authors conducted a mediating randomization analysis to address this cause effect. And they found that DNA methylation alterations are predominantly the consequences of adiposity rather than the cause. They also found that many of those genes are involved in lipid metabolism, substrate transport, and inflammatory pathways. Interestingly, obesity-related DNA methylation marks predicted future risk of type 2 diabetes. So this study provides strong evidence that obesity has a powerful influence on epigenetic modifications across the human genome. There is evidence that DNA methylation can predict future risk of chronic diseases, including cardiovascular disease. We recently conducted a genome-wide analysis of DNA methylation and the risk of acute coronary syndrome, or ACS. We used a two-stage design in this study. In the discovery stage, we identified almost 200 CPGs with differential methylation in blood between the ACS cases and the matched controls. In the replication stage, we were able to replicate 47 CPGs associated with ACS after adjustment for multiple testing. Those associations were independent of blood cell types, blood lipids, and other cardiovascular risk factors. In further analysis, we found that associations identified in the whole blood were mainly due to methylation changes within certain immune cell subtypes. So this study provides evidence to support the role of inflammation and immunomodulation in the pathogenesis of cardiovascular disease. It's well established that DNA methylations are strongly correlated with age. So the question is, can epigenetics be used as biomarkers of human aging? There is promising evidence that DNA methylation can be used as a novel and a reliable biomarker to assess biological aging and predict longevity. In the past several years, DNA methylation age predictors have been developed. The first age predictor is called Harlem age predictor. In 2013, Harlem and colleagues developed a methylation signature with 71 CPGs that predicted true age with excellent accuracy. The age predicted by those methylation sites was called methylation age. Methylation age can be derived as either aging rate or delta age, which is the difference between the predicted methylation age and the true age of the participants. This figure shows a very strong correlation between the predicted methylation age and the chronological age of the participants. The individuals above the diagonal line had increased methylation aging, whereas the participants below the line had reduced methylation age. The second methylation age predictor is called Harvard's age predictor. Also in 2013, Steve Harvard developed a methylation age predictor using 353 CPGs for multiple tissues. This age predictor is highly accurate with a very strong correlation with the chronological age. The correlation coefficient is 0.96. The author stated that DNA methylation age measures the cumulative effects of epigenetic maintenance system. This novel epigenetic clock can be used to address a host of questions in developmental biology, cancer, and aging research. In the past several years, methylation age has been linked to a wide range of dietary, lifestyle, and environmental exposures, and also various health outcomes, including type 2 diabetes, cardiovascular disease, cancer, and mortality. Several studies have shown that DNA methylation age acceleration can predict future risk of death. This is a meta-analysis of 4 cohorts of elderly participants. In this study, methylation age acceleration was calculated using both the Harlem and the Harvard's age predictors. Methylation age was found to be moderately heritable with heritability coefficient of 0.43. This study found that increased methylation age is associated with increased mortality. In particular, 5-year increment in the hormone delta age is associated with a 16% increased risk of mortality. These findings highlight that methylation age is a valid aging biomarker and can be useful in future studies of aging. Emerging evidence suggests that diet, lifestyle factors, and metabolic conditions can influence methylation age acceleration. This is a cross-section of studies of more than 4,000 postmenopausal women in the Women's Health Initiative. This study calculated methylation age acceleration using blood DNA methylation levels based on the Harlem and Harvey's age predictors. This study identified several diet and lifestyle factors associated with methylation age accelerations. In particular, unhealthy diet with decreased consumption of fruits and vegetables and fish was associated with increased methylation age acceleration. Also, decreased physical activity, increased BMI, and increased waist to hip ratio were also associated with increased methylation aging. Meanwhile, several metabolic parameters, including increased triglycerides, increased blood pressure, decreased HDL, and increased insulin and glucose, were associated with increased methylation aging acceleration. So this study provides promising evidence that diet and lifestyle modifications can slow down or reduce methylation age acceleration. In summary, a healthy diet and lifestyle is the key to healthy aging and longevity. Diet and lifestyle factors have epigenetic benefit by influencing DNA methylation alterations and the gene expressions and the gene functions. The epigenetic aging markers have been associated with mortality and age-related diseases. There is promising evidence that a healthy diet and lifestyle may reduce methylation aging. In terms of future research directions, we need more longitudinal studies with large sample sizes and more relevant target tissues to analyze methylation marks. We also need more research on clinical translations for disease prevention and the treatment. So how to turn back or reset your epigenetic clock? Certainly there is no magic bullet to do that. Current evidence suggests that a healthy diet and lifestyle has significant benefits in terms of reducing risk of chronic diseases, improving longevity, and slowing down or turning back your epigenetic clock. Thank you very much for your attention.
Thank you so much, Dr. Hu. That was really an interesting program on epigenetics.